Now we're coming to the last part of the presentation about our project this year. And in the end, I will quickly talk about recruitment for next year's team and how to join. You might have seen me before. I was the guy that did the advertisements in the lectures. I'm also taking care of the recruitment for next year. And I was a member of this year's team, particularly for human practices, so outreach and talking to experts and finding a place in the world for our project. And one thing you will notice, the previous presentation was really, or the previous project was really a lab project with a focus on the lab work. What I will present you now, our project had a lot more to do also with the lab, and actually in hours spent, the lab is still vastly the majority. But in presentation, what we have, we really went outside of the lab to go and talk to people, to get interactions from the outside, to get expert knowledge on how our project could really affect the world and how to improve it so that it could do an even better job than it originally envisioned. So first, quickly, what is the goal of our project? We worked with these plant pathogen recognition receptors, also known as PRRs. If you haven't heard about them yet, that's fine. I will give an in-depth or more in-depth introduction of them later. For now, they are membrane receptors that are part of the plant's immune system, so they detect pathogens. These PRRs are extremely important in agriculture for transgenic resistance in plants, so they're being used a lot. But as of right now, nobody has ever thought to use them in a biosensing system, so in a system that uses these PRRs as receptors to detect bacteria, but then give an output from it that we can then read and interact with. As such, we wanted to lay the groundwork for these PRRs, and we wanted to create a proof of concept project. So we wanted to make an organism, yeast in our case, that uses these receptors and actually gives us an output to the presence of microbial contamination or bacterial contamination specifically in our case. There you see the PRRs, but as mentioned, I will go into more detail later. Because first, I would like to start a bit where we started and go back to our inspiration. So before we had any idea what our project was going to be, what our lab was going to be, we just wanted to do something with biosensing. We thought the idea of using an organism to detect environmental issues was an extremely cool concept and had the advantage that you could make systems that were extremely cheap and simple that could give outputs that are incredibly complex to detect with current technology, because biological organisms are a lot better at working on certain things on a microscopic scale. That's why they're way better at producing insulin than we are, for example, because we have to go through multiple steps and processes that for biological organisms just come naturally. So when we then actually had to switch labs a couple times and we ended up with the Institute of Plant and Molecular Biology in the lab of Professor Cyril Zipfel, we created our project because he's an expert in these PRRs, and we wanted to do environmental sensing, biosensing, and it just kind of added up. That created the project to make a microbial sensor in water. Originally, we really wanted to lay on the strengths of biosensing, which is that it is extremely cheap and simple to use because it does not require machinery or any other equipment. And because of that, our original idea was to make something that really anyone could use anywhere. To have a colorimetric output, that means you can see it with the naked eye, and that will give a readout to bacterial contamination in water that was yes, present, or no, absent. But to really, find how your project affects the world and how you can improve it so that it really has an application that's actually useful, you need to go out and talk to experts. And that's exactly what we, or specifically, that was my job in the team, did. And during that, we found a lot of issues with our original system and ways in which we can improve it. So we talked to experts in biosensing, microbial sensing, and even people that work, in developing nations on water safety from the WHO. From that, we kind of have 3 key points that made us change. Yes? Okay, yeah. 3 key points that made us change how we looked at our project and how we actually designed on a molecular level our system. So first, this idea of working with laymen, with general people with no experience in microbial sensing, sounds nice, right? Anyone can use it anywhere, it's super cheap. But as it turns out, that's very difficult to actually do because the kind of information you can provide to a layman is very specific and limited to the kind of information they can work with and do something with. In our case, and I'll get to that in the system, why it is like that, detecting all bacteria, so a total bacterial load of a sample, is information that's actually very difficult to use if you're not looking at it on a distribution level, on a level of multiple samples, which is why our kind of information actually is very difficult for a layman to process. Also, information is not useful, especially for a layman, if it does not lead to further decision-making. And one thing we learned is that in almost all contexts, it doesn't even matter that much whether the water is contaminated, because chlorination, almost everywhere in the world, is such a simple solution that if you're not quite sure whether the water is contaminated or not, then you can just chlorinate it and it'll be fine. And we cannot deliver a system that gives, a powerful enough and informative enough output to decide we will not chlorinate this system. The dangers of, having a false positive are just too large. That's not something we decide or our system can provide. The second thing is we learned, okay, layman doesn't work, but experts, so laboratory personnel, people that work in water safety, we talked to them and they actually have a lot of things that are currently limited with their sensing. And 2 points that really come out are selectivity. So finding bacteria is easy. Finding specific bacteria is a lot harder. And the second thing is the current really cheap systems, which is what the market we are competing with, right, have a lot of limitations when it comes to their speed, their accuracy, their sensitivity, or their specificity. No system currently covers kind of the same niche that we do. And that's how we adapted our project to really fit into a niche that no other currently existing system fills. And the last thing is something we really didn't want to work with that much. It's something, when you work on a synthetic biology project, you kind of move it to the back because it's a problem that every synthetic biology faces if it produces something like this. We are producing a GMO, a genetically modified organism. And originally, at least, we intended to hand it to laymen. That obviously faces the issue, how do we prevent the escape of the organism? How do we stop this organism from spreading in the environment with our edits in its genome? And that is something in the end we decided we cannot guarantee. And therefore we had to say, we create a new project goal. We are still making an ultra rapid, cheap, and super simple system, but it is now supposed to be used as a supplement to laboratory systems. And it now doesn't give a yes or no output. It gives an output that is quantifiable. That means you can really say how much bacteria is in a sample. Now, for that, we of course had to give up this colorimetric aspect that visible to the naked eye. We now need a luminescence meter to read the output, but that still makes it a lot more portable and rapid than comparable systems. And of course, advancing the use of PRRs is not something we looked at in our human practices, in our outreach, but is something that is still very fundamental to our project. And all of this is how we designed, or what influenced the design of our system that we'll get to now. But first, I quickly need to explain PRRs. And I'm a bit delayed on time, so I'll speed it up. But generally, PRRs are a huge family of membrane proteins in the plant innate immune system. That means they are genetically inherited, they don't change throughout the life, and they detect extremely conserved bacterial epitopes, so parts of proteins, actually from all microbes, but the ones we chose are bacterial. Why are these really interesting for microbial sensing? Is because they have this, there's so many of them, and they each have such a broad range of recognition that there really is a huge amount of options with the system based on them. And because they are biochemically similar, they all kind of have the same structure, we believe that if you can express one of them, so prove that one of them works as a biosensor, then the others, or at least most of the others, should function as well. There's also a few further points to future development of PRRs that I will get to later. So the three PRRs we chose are called Ephelis-2, Core, and EFR. These recognize, for example, flagellin, which a huge number of bacteria have, and the elongation factor necessary for bacterial replication. So the idea was, A, these are well-studied, well-known PRRs, which for us is very important. We do not have the ability to go, all the way back and find a new PRR. But these are also PRRs that recognize extremely broad spectrums of bacteria. And in fact, the combination of these three, we can relatively safely say would recognize every bacteria possible in a water sample. So the idea now is we take these PRRs, these three that we have chosen, and we put them into yeast. And now all we need to do is get an output from this system. We need to somehow be able to read out how many bacteria there is in a sample. And for that, we use this property that these PRRs have, which is they dimerize in the presence of a bacterial epitope. So here you can see two receptors, for example, this EFR receptor and its co-receptor. And what we did is we attached half of a luciferase protein to each of these receptors. Now, if a bacterial epitope is present, you can see here, this red thing is a part of a bacterial protein. These two receptors come together, they interlink in the presence of a bacterial epitope, and with that, the luciferase at the bottom of these two receptors fuses again, becomes fully functional, and starts glowing. And that is the readout we can then take with a luminescence meter, because the more bacterial epitopes present, the more of these luciferases will be activated, and with that, at least we hope you can quantify the amount of bacteria in a sample. Now, I just quickly want to show this. I don't think you can or should read any of this, but our system is incredibly simple, right? We're not using 20 different parts to do what we do. Really, the core of it is only these two parts, the receptor and the luciferase output. But of course, we need to add, promoters, signaling proteins to get them to the correct location in the cell, and so on. But in general, with two of these plasmids, one for the main receptor and one for the co-receptor, you can build our entire system. Now for our results. Our results were not quite what we had hoped for at the beginning. So we had a lot of difficulties, finding a lab during Corona, having the amount of people in the lab, but also we were all inexperienced. And that is, inexperienced at the start of the iGEM. And that is something that I kind of want to stress if you intend to participate, is we were not experts when we started this, right? If you have an interest and a motivation to learn, then you can take part in iGEM. But the result of that is, of course, that sometimes you will have lofty aspirations that you then cannot realistically reach, and you will have, a lot of setbacks in between. Now, to our results. The first result I'm showing you here is the result of a confocal microscopy. And now you can't read it because this thing is here. So here you can see two images. On the left, this is a confocal microscopy of a wild type yeast. As you can see in the top image, there is no fluorescence. That's how the microscope is adjusted. On the right, you can see yeast cells that have our BAC1, the co-receptor, integrated into them with a YFP attached to it. So the idea of this result is that wherever there is a yellow dot, there is a yeast that has a YFP, and where there's a YFP, there's a receptor. Now, unfortunately, there's two issues with this. A, the resolution here is not quite good enough, and we had too much fluorescence as that we could identify localization in the cells. So maybe if we look at the, I do not want to go on. Okay, if you look, this is the exact same image, just slightly larger. You can see that the entire cell is covered in fluorescence, which means we cannot identify whether the receptors are only at the membrane or spread out across the entire cell. Now, later on, we even noticed a further issue with this image, is that we are not quite sure if this is actually fluorescence from the YFP, or if we had issues setting up the confocal microscope properly, and this is actually still autofluorescence from the yeast. So because of that, we are still running further tests actually tomorrow and as of right now, which is something, you know, the deadline is 27th of October, 2 weeks from now. You really probably want to avoid, but at the end you saw the previous team did it, we did it. It might very well happen to every team that at the end, you know, you still want more results. The second results we got were this Western blot. And in case you're not aware, a Western blot divides proteins by their size and then stains specific proteins, right? So what you can see here is a Western blot of three things: untransformed, Kurer, so one of our receptors, and EFR, one other of our receptors. And it is stained with this alpha flag to show, you know, the GFP, or in this case, the RFP we attached to these receptors, are being stained with this alpha flag. And what you can see is there is two lines on this Western blot, one at about 115 and one at about 37 or 38 kilodaltons. So that's the size of the protein. This is very good because Kurer, the receptor we want to see here, has a weight of 80. But because it gets glycosylated quite heavily after production, it is actually a very good assumption that this 115 result is actually our Kurer receptor, right? Now, the lower one is maybe our BUC1. So BUC1, the expected weight of that protein is 39 kilodalton. And as you can see here, it's below 39. Now, it should theoretically be even higher than 39 because as the Kurer, it also gets glycosylated. But it is also very difficult to read out very exact values on a Western blot like this. So we are not entirely certain if this is actually our BUC1 or if this is maybe a degradation product of our BUC1. Now, you might also see that there's no staining at all in the EFR lane. And once again, these results are a bit more extensive than what I just shown you. So there's two issues here. On the left, you can see two Western blots, the one I just showed you and a completely empty one. And that is actually because the staining we used stains only half of the split protein. And it turns out we used the same split protein twice. So you're supposed to use A and B, and we had B twice. But that is fine because it's just a result to show that it's present, right? In the final result, theoretically, you would want to see, you know, one receptor with the alpha flag and one with the alpha HA. The second thing, the issue with not being able to see the EFR, you can see on the picture on the right. The picture on the right is a stain of all proteins, all of them. And as you can see, it's not just the EFR receptor that isn't really present in the EFR sample. It's actually almost all proteins that are not present. So that must have been some issue with the production of this result. And that is actually it so far for our laboratory results. We're still working on it. We still believe to have some more results coming in. But as you can see, our project has a lot more to present in its, you know, outreach, in its how we looked at how to actually implement it than the lab. And that is something that's important for iGEM as well. You can have projects that are almost entirely in the lab, such as last year's. Or you can have projects that are a lot more on the outreach, on the implementation side of things. And as mentioned, some not even in the lab at all, purely modeling. So I've talked about it before, and I'll keep it really short. The idea of our system really is, we put our yeast in a sample tube, we hit it with contaminated water, and we throw it in a luminescence meter. And from there, we can more or less directly read out the quantification in a sample. The last thing I want to say is that these PRRs, why they are so interesting to us is not just their properties that they currently show, but also because they are being researched so heavily in relation to agriculture there's actually so many more of them that are known with specificities that might be very important for microbial sensing, such as, for example, viruses and protozoans, which are currently incredibly difficult to identify. And currently there is some receptors that identify viruses, but more importantly, people are actually making engineered PRRs, so designing them themselves with a specific affinity to a specific bacteria. If that succeeds, that would of course be fantastic for a system like ours, because in theory, all you have to do to use a new receptor in your system is to exchange the receptor in the plasmid. And there's of course, right, once you have that, there's also a lot of options with outputs and other things you could add, such as, for example, a threshold. So there's a lot of modularity possible in this system. So last but certainly not least, I just would like to show you my team. Some of them are here with us today, and I'll ask them to come up on stage in a second. You can ask them about iGEM. You can, ask them about how it is to take part. And you can ask me specifically about recruitment together with Sophie and, where's Martina? Oh, there's Martina. About recruitment for next year. Quickly, our lab, as said, is the Zipfel group, together with our personal instructor, Cyril Zipfel. You might have seen him in the plant biology lecture, and our two instructors that were really with us in the lab and helped us, and without them, it wouldn't have been possible, Kyle and Isabel, and of course, all of our sponsors that helped us make this project possible financially. Something that I've kind of stressed in this presentation is, in contrast to the previous team, a lot of out-of-lab things going on. And a couple other things I would just like to show off here is, once again, we did this Swiss iGEM meetup, this time with EPFL and UNI Lausanne. But one thing that really gives you flexibility in iGEM is the gold medal. So you saw silver, last year's team achieved silver. The gold medal is different from silver, where it does not require you to fulfill all requirements for silver, but instead gold lets you pick 2 out of a wide list of requirements. We chose, for example, education and communication, part improvement, and integrated human practices. I know it's 3, you only need 2, but we went with an additional 1 to, be on the safe side. And with that, really, right, education and communication, we went to classrooms, we organized a workshop. Those kind of things are also part of iGEM. Also, this year, special for this year, we had to make videos. We made this really cool video showcasing our project in 2 minutes that you can watch on the iGEM video page together with videos of all the other iGEM teams. And we also need to make a project presentation purely in video form this year. So that is all for our team this year. I would just quickly like to say a few words for the recruitment, if you're actually interested in joining next year's team, or at least considering it. So first off, just quickly, the recruitment process. There's 2 steps to this. First, until the end of October, you can send in an application form. This is really just a broad questions form, and our goal here isn't to, weed out participants. We just want to get to know you a bit, so that then when we invite you to talk to us, to get to know you a bit, on a more personal level, to see who we think is really suited for the team, so that we can then kickstart next year's team. And this is something that's important to us. We are not organizing the team in the sense that we are telling you what to do. We want to kickstart the process by selecting the core members of next year's team, right? Getting you together, getting you to know each other. But after that, it really is up to next year's team to self-organize, to find out who takes what roles in the team. And if you want to, who else is part of the team? We took up 2 additional members way after the recruitment phase in our team, and we're really glad that we did, right? That is an option. But also, iGEM is a huge, huge time commitment, right? It takes up most of the summer holidays with work and a bunch of work during the semester, both before and after, right? Now, for the core team, that is also what we expect, right? This project is something huge, and you need to be able to give a lot of time to it. But if you're sitting here and you're currently considering, I'm really interested, but I simply cannot give that kind of engagement, maybe you just want to talk to the new team and see if you can maybe fill, a more minor role, if you can participate without, being part of the core team that really spends their entire summer. One thing that we have, that we kind of focus on this year, is that we don't want to divide the team into these super strict roles of, this is a lab person, this is a human practices person, this is a website design person, right? We've really focused on this approach where we want you to tell us about what interests you and what fields you want to work in, what fields you have experience in. And you'll see that on the open questions formulary. We just want you to tell us a bit about what interests you and what you're willing to work with, so that we can then kind of build a team that covers the basis for iGEM. From there on out, it is the team's own responsibility to find other members if they think they are necessary and to organize internally. That is, of course, not to say that we are just going to abandon you. I and a bunch of this year's team, last year's team, SynBio advisors that have been in iGEM multiple times, everyone is still available to help you. But it really should be your project that you can design yourself. And we're not here to tell you what you can and can't do. We're just here to help you after kickstarting the initial process. And that is all from me. Thank you very much, all of you, for listening.